Dr. Baliga here. I got another terrific God Knowledge Doc podcast today for you all. This podcast series discusses current day medical topics. Today I have a terrific podcast for you. It's titled Transient CAR-T Cell Therapy for Cardiac Ventricular Dysfunction. It's from the seminal paper which was released online on Science in December of 2021, and the hard copy was available in the first issue of Science, 7th January 2022. It's titled CAR-T cells produced in vivo to treat cardiac injury. There are 4 senior authors for this paper, including Dr. Haig Agayanian, Dr. Drew Wiseman, Dr. Hamida Perhiz, and Dr. Jonathan Epstein. I want to thank Dr. Jonathan Epstein for sharing his slides with me for this podcast. I had the fortune of working in Dr. Tom Smith's lab at the Brigham and Women's Hospital and Harvard Medical School in the Thorne Building, and Dr. Epstein's lab was across the hall from Dr. Smith's lab. Cardiac fibrosis in cardiac dysfunction is a consequence of excessive deposition of extracellular matrix protein by activated cardiac fibroblasts. Current therapy, as you all know, including RAS inhibitors, including ACE inhibitors, ARNIs, and mineralocorticoid receptor blockers, which limit progression of fibrosis rather than the added fibrosis which is once established. This paper in Science by Dr. Epstein and collaborators at UPenn describe a novel approach to attenuate activated fibroblasts by combining the properties of engineered T cells, that is CAR-T cells, with those of mRNA therapeutics. These investigators deploy CAR-T cells in vivo to target activated cardiac fibroblasts, attenuating interstitial cardiac fibrosis. One advantage of the COVID pandemic is the field of mRNA therapeutics came front and center, and these investigators worked together to deploy this technology to help patients with cardiac fibrosis. This is an animal study. The expression of CAR, that is chimeric antigen receptors, on T cells is transient in this study because mRNA is intrinsically unstable and no CAR-T cells persisted beyond a week. Thus, this T cell targeted mRNA platform allowed reprogramming of T cells in vivo and did not require removing T cells from the patient, which is an approach that circumvented the costly and risky infrastructure that is required for CAR-T cell generation. As you all know, CAR-T cell is used for hematologic malignancies. Fibroblast activation protein is a cell surface glycoprotein. It's a gelatinase in the serum protease family. It's expressed during embryogenesis but not in adult tissues unless injured or when there's cancer. It includes activated proliferating fibroblasts. 90% of the tumor stromal fibroblasts contain FAP. It's apparent in the human myocardium after acute MI, after acute myocardial infarction. What did these investigators do? They performed a series of in vitro and in vivo experiments. Initially, they performed the in vitro experiments. They generated mRNA encoding chimeric antigen receptor against the FAP, that is the fibroblast activated protein. This mRNA was then packaged in lipid nanoparticles, LNPs, that targeted CD5, which is naturally expressed by T cells. The study investigators found that with this approach, that is the CD5 targeted lipid nanoparticles, delivered the mRNA to most of the T cells in culture, that is about 83% of the T cells in culture. The chimeric antigen receptor expression peaked at 24 hours and then rapidly decreased over the following days, which is about a week. This newly generated CAR-T cells effectively eliminated fibroblast activated protein expressing target cells in a dose-dependent manner. Next, they assessed the abilities of these CAR-T cells in vivo. They utilized a hypertensive mouse model of cardiac fibrosis induced by constant infusion of angiotensin II and phenylephrine for a week. The study authors injected intravenous lipid nanoparticles, LNPs, carrying the fibroblast activation protein, CAR, that is a chimeric antigen receptor mRNA, or FAP CAR-mRNA. 48 hours after the injection, the authors were able to detect a consistent population of FAP CAR positive T cells, about 17.5 to 24.7%, and the expression of FAP CAR was transient. It was about a week. In this figure, you'll see that when the mice were treated with an infusion of angiotensin II and phenylephrine, there was significant fibrosis by the reddish staining of the mouse heart, and FAP was expressed by fibroblasts of the mouse after 1 week of angiotensin phenylephrine. This slide nicely gives the time sequence of their experiments in mice. The pump was inserted in the mice on day 0, and the mice were infused with a constant infusion of angiotensin II and phenylephrine to induce cardiac fibrosis. A week later, once the fibrosis was established, the lipid nanoparticles carrying the FAP CAR-mRNA were injected intravenously. The study investigators were then able to detect a consistent population of FAP CAR positive T cells 48 hours after the injection. That's in about 17.5 to 24.7%. A week later, they found that the expression of FAP CAR lasted around a week later, underlining the desired transient effect of the approach. After 2 weeks, that is day 28, the authors observed significant improvements in cardiac function in treated mice. LV diastolic function returned to uninjured levels, while LV systolic function also considerably improved. This slide shows the CAR-T cells populate the scar. So when you look at the figure on the right side, they found that after 2 weeks there were significant improvements both in LV diastolic function. In fact, the LV diastolic function returned to uninjured levels, and the LV systolic function also considerably improved. And if you look at the echo parameters, you can see again that there were substantial improvements with FAP CAR-T cell therapy in cardiac function. In fact, if you look at the middle panel, you can see that in the controls there was no change in the diameters of the LV. When they stained the mice or the heart of the sacrificed mice, they found that in vivo FAP CAR-T cell therapy reduced cardiac fibrosis after injury, and when they charted it, you can see that there was considerable improvements in cardiac fibrosis. Histologically, there was a significant reduction of the overall burden of the extracellular matrix. And compared to the middle panel, which is mice treated with angiotensin II and phenylephrine, you can see in the third panel there was substantial reduction in cardiac fibrosis with FAP CAR-T therapy. Histologically, neither the lymphocyte B or natural killer cells expressed FAP CARs. No histological changes in non-cardiac organs were noted, nor was there any weight loss. These findings suggest the safety and the specificity of this approach. So their studies suggest that this therapy appears to be safe in mice, and it has been reported in humans with limited reported toxicities. We need larger studies to confirm these findings. So in summary, engineered T cells can be used to treat pathologic activated fibroblasts expressing fibroblast activated protein. T cells can be engineered in vivo using targeted mRNA with lipid nanoparticles. Unlike in cancer, there is no requirement for memory or complete killing. Reduction in fibrosis should be beneficial even if not complete. The next step would be to evaluate which fibrotic diseases might be amenable to such therapy. And another important next step would be to study large animals and eventually patients. This slide shows that the human cardiac fibroblast gene expression, so there is a range of fibroblast genes which can be targeted. And finally, you will see from this very nice perivascular fibrosis staining that even though the therapy reduces interstitial fibrosis, there remains substantial perivascular fibrosis. Presumably the genes which are responsible for perivascular fibrosis are somewhat different from the interstitial fibrosis genes. This study is exciting. It's going to be a game changer. In fact, it'll be really interesting to see whether targeting different fibroblast genes will reduce fibrosis in other organs, and if they're able to reduce perivascular fibrosis, then we could even improve hypertension. As you all know, as we age, the vascular system becomes stiffer, and if we could give engineered CAR-T cell therapy which are transient, we could potentially reverse hypertension. This paper is a game changer. I'd like to thank Dr. Jonathan Epstein, one of the four senior authors of this paper, for sharing me his slides for this podcast. This paper is a game changer. It's going to change therapeutics once it's possible by the bedside, but it's an outstanding paper. In a nutshell, CD5 targeted lipid protein nanoparticles deliver therapeutic mRNA to the lymphocytes in vivo, where they form transient anti-fibrotic chimeric antigen receptor T cells, which then significantly improve cardiac function in the mouse model of heart failure. This technology enables CAR-T therapy to be therapeutically dosed and mass manufactured, eliminating many current barriers to broad CAR-T use. In fact, this is like a micropharmacy which you can infuse and target it to the organ of interest. I would like to thank Dr. Aarti Asnani, MD, cardiologist at Harvard Medical School and Beth Israel Hospital, for bringing to my attention the importance of this paper. This ends the podcast. These are the books which form a background to many of my podcasts. Follow me on Twitter and let me know what you think about these podcasts.